Here, at UCL, situated in the heart of London, researchers have been studying the mechanisms underlying the regenerative capabilities of the peripheral nervous system. In the research labs at the MRCL-MCB at UCL, the research team led by Professor Alison Lloyd have uncovered novel cellular processes by which an established tissue can be repaired within an adult organism.
We're interested in tissue regeneration. Like other mammals, we have a very limited ability to regenerate tissues following an injury. This is in stark contrast to other complex organisms, such as types of amphibian like the newt, which can regenerate whole legs or tails following amputation. However, within our bodies, a few of our tissues have retained impressive regenerative capabilities, and one of these tissues is the peripheral nervous system. And by studying how this system regenerates, we hope to gain greater insight into the cellular and molecular mechanisms which underlie tissue plasticity and organ regeneration.
Peripheral nerves connect limbs and organs to the central nervous system. The axons of the nerve are surrounded by a second highly specialized cell type, glia, known as Schwann cells. Following the cut of a nerve, the stump retracts and the axons downstream of the cut degenerate. Inflammatory cells, together with dedifferentiated Schwann cells, then bridge the gap and the axons regrow across this bridge back towards their targets. However, this requires the axons to accurately find their way. To date, the mechanisms have been unclear.
Looking at sections of that nerve following a cut, we found that by 2 days the ends have rejoined, forming a bridge consisting of cells other than Schwann cells, which are labelled green. By day 5, however, cords of Schwann cells can be seen streaming out of both stumps, and axons, which are labelled red, are growing along with the cords of Schwann cells on the way back to their targets. When we looked more closely, we could see the Schwann cells in clusters, with the axons on their surface apparently following behind the Schwann cells. However, clusters of other cells which we found were fibroblasts were surrounding the Schwann cell clusters but not commingling, suggestive of a cell sorting process. We found that this cell sorting process could be mimicked in vitro, and when we videoed the cells we could visualize the mechanism underlying this behavior. In these videos, the green Schwann cells exhibit a behavior known as contact inhibition of locomotion, whereby certain cell types repulse each other, behavior which leads to an even distribution of cells. However, when we mix the Schwann cells with the fibroblasts, we see a switch in cell behavior, in that when a Schwann cell contacts another Schwann cell, it is no longer repulsed but adheres to it, resulting in the formation of clusters. So, fibroblasts switch Schwann cell behavior from repulsion to attraction.
The next step was to determine the molecular mechanisms underlying the switch in cell behavior. An important clue came from experiments in which we treated Schwann cells with purified fibroblast membranes and found that these were sufficient to cluster the Schwann cells. This indicated that direct cell-cell contact is required for Schwann cell sorting. We found that Fs and ephrins are the molecules responsible. Specifically, we show that ephrin B ligands on fibroblasts signal to FB2 receptor on Schwann cells. Downstream of FB2, we discovered that upon receptor activation, the transcription factor SOX2 mediates cell sorting by inducing the relocalization of the adhesion molecule encadherin to the Schwann cell surface. This makes the cells stick together and form clusters, explaining how fibroblasts can switch the behavior of Schwann cells from repulsion to attraction.
To address the importance of this process in nerve repair, in collaboration with Ralph Adams in Germany, we performed nerve injury experiments in FB2 knockout mice and saw a dramatic difference. In controls, axons grow out in a directional organized fashion. In contrast, in FB2 knockout animals, we saw axonal outgrowth was disorganized and the directionality was lost.
These studies describe a novel role for fibroblasts in wound healing, in that they orchestrate a repair process by direct cell-cell interactions. Schwann cells, upon dedifferentiation following injury, switch on both FB2 and SOX2 and are thus primed to respond to the fibroblasts. However, it is only at the wound site, where there is an influx of fibroblasts, that ephrin B signaling sorts the Schwann cells into cords, initiating the coordinated migration which takes the axons back to their targets, ensuring efficient nerve repair.